Welcome everyone to this session now on Biodiversity Genomics Europe. So my name is Peter Holland and I'm chairing this together with Kerstin and Howe. And the goal of this session is we want to showcase some of the work done on genomics, biodiversity genomics within Europe and using and analyzing European species, but a combination of those two. So what we have is 6 shorter talks, and you've got, hopefully on your screen in front of you, you can see those, you can see the titles of those. And we're going to have 3 and then questions and then 3 more talks. So just to sort of set a bit of the scene for this, I want us to have a little think about what's happened to genomics over the past 50 years or 70 years, really. And I like to think we're seeing huge jumps, really, in sort of intervals of about 20 or 25 years. And of course, lots of things happen in between, but just see that what's happening in each of those huge jumps. So from the proposal of a double helical structure of DNA way back in 1953, then jump forward 20 years to the invention of DNA sequencing, jump forward another 20 or so years to the first complete genomes of complex organisms, including human genome. And of course, even those were met with enthusiasm around the world. You see these quotes here from Bill Clinton and Tony Blair. At the time, that was the time of the human genome being released, and so much excitement about that. But where are we now if we jump forward another 20 years? How would we characterize genomics in 2024? And I think really there are 3 big things that have been happening. The first is, of course, the obvious one, which is numbers. We've seen, and you've just heard in the talks from Harris Lewin and Mark Blackster, the phenomenal numbers of genome sequences coming out around the world. Or increase in the output is something that's quite striking. But there's also been a shift really in terms of openness of the data. So now some of the large genome sequencing projects, for example, the Darwin Tree of Life and several others, have become committed to releasing these genome sequences immediately and open without restriction. And I think that's a real change in approach to do science, as opposed to if you go back maybe even 5 or 10 years, where a group or a consortium would sequence a group of, then analyze it and then publish. This model has been flipped. So the data of many, many genomes are seriously available for the whole world to analyze. And the third point I'd put there is inventive new uses and applications of genome data. We can even forecast where that line is going. And I think we'll hear some of that today. And I think of that again in 2 different ways. We have what we might call direct analysis, where the genome sequences themselves are an absolute treasure trove of information, all by analyzing and browsing those genomes. But of course, the genome sequences also enable lots of analysis, further analysis, for instance, variation within species, gene expression patterns, etc. So let's return to what we're going to hear about in the next course. As I said, we're going to have 3 talks from Saad, Arif, Claudia, Martin, and Camille Cornet, and the titles of their talks are there. Please put questions for them in the Q&A box on the Slido, and then we will see those questions, break after the first 3 talks and ask some of those questions, and then we will reconvene immediately and carry on with the next 3 talks. So that's it by way of me, and I'm going to pass on straight. You will hear pre-recorded talks from the speakers, but the speakers will be live here. They are live here, and they will be able to respond to your questions live and on screen. So we'll hand over to the talks now.
I'll be talking about our recent work on genomic erosion in the extirpation of the black vein white, or Aporia crataegi, in Britain. For those of you not familiar with the species, the black vein white butterfly is a large and common butterfly. It ranges from Northwest Africa all the way to Japan, and in the north up to Lower Scandinavia and Siberia. Previously, people have looked at CO1 mtDNA haplotypes, and there's a few major haplotypes scattered throughout its range, but they're largely dominated by one main haplotype. In Great Britain, this was once quite a common butterfly. You could see them in large numbers. However, it disappeared in the 1925, around the 1925s. More recently, it's also been extirpated in Netherlands and Korea. Its demise in Great Britain has been described as spasmodic rather than gradual. This is because there was a decline in observations starting from its northwestern range in the mid to late 19th century, all the way to around 1925, where the last observation came from Kent in southeastern England. During the 19th century, there were already alarm bells being raised about its disappearance, and there were already several releases from European stock. Many of these seem to be larvae imported from Germany and then released into Great Britain. There's been much debate about the reasons behind the demise of the black bean white in Britain, but some of the most convincing of these include avian predation. There was an increase in small birds in the 19th century, presumably due to a decrease in raptorial birds. Pathogen infection has also been implicated, potentially fungal or viral. And additionally, there does seem to be some correlation between some extremely wet Septembers around years of low black vein white abundance in the 19th century. It's quite likely that's a combination of these factors, and perhaps additional ones led to its eventual demise. But the wet conditions and a combination of fungal infection has often been cited as one of the main causes. What we wanted to do was provide a fresh genomic perspective on the demise of the black vein white in Britain. We did this by sequencing whole genomes from black vein white butterflies prior to 1925 from the UK, but also from Europe. And what we wanted to do with this data was to address how the effective population size of the black vein white changed around and after its colonization in Britain. And also if we could find further evidence of population size reductions in the 19th century, and also to see if there was a decline in individual genetic fitness of the Great British populations. And finally, this is still ongoing work, but we also wanted to do some metagenomics to explore whether there's any evidence for pathogen infection in the extirpation of UK butterflies. In the results we present, we also present a complementary analysis on the common blue butterfly, the Polymetis icarus. This is a demographically stable species in Great Britain, at least in recent times, but shares a similar post-glacial colonization history to the black vein white. We sample museum specimens only where collection date and locality was available. There were many more specimens in the museum archives, but in many cases, this information wasn't available. We made libraries in-house from our historical DNA. We did this using a double-stranded DNA library prep kit, which makes use of the user enzyme, which leads to partial repair on the 5' ends of our historical DNA. We also only used 1 to 2 legs per butterfly as template DNA because in our hounds we found we got more endogenous reads from libraries made from legs rather than those made from abdomens. Our final samples of the black vein white range from 1854 to 1924. The average depth of European samples was around 10x. The average depth for Great British samples was around 6x. As expected, the DNA damage profile at our 5' ends isn't characteristic of what you would expect from historical DNA. This would have much higher C to T transitions, which is this red line here. However, on the 3' ends of our reads, we do find characteristic DNA damage of G to A transitions, which is this blue line here. In contrast, our common blue data is of fairly high quality because these are recent specimens. The depth ranges from 11 to 50x, but the sample sizes here are fairly small. To give you sort of a spatial idea of our sampling, this here, the blue symbols are samples from Great Britain, the orange ones are from Europe. The triangles represent common blue samples, and the circles represent black vein white samples. So we did a PCA of genotype likelihoods of our black vein white data, and what we find is a tight clustering of the European samples here. There is more disparate clustering in the Great British samples, but this could presumably evidence of isolation by distance, as these samples here are all from the southeast, and these samples here represent the western part of its range. We also found 3 Great British samples that clustered on the other side of our European samples. These are presumably European stock releases into Great Britain, rather than a case of mislabeling. And for downstream analysis, we do label these specimens as European to bolster our sample size. However, our conclusions are robust to whether we include these specimens or not. Interestingly, the youngest specimen in our sampling from Great Britain, which comes from Kent in 1924, clusters with European samples rather than Great British samples. We reconstructed historical demography using PSMC for both species, for an individual from Europe and from Great Britain, using individuals with the highest coverage we had available. Here we have effective population size on the vertical axis and time on the horizontal axis, with more recent times closer to 0. We have black vein white on the left and the common blue on the right. And the thing that's common to both these plots is this exponential increase in the European populations around the time of the last glacial maximum, which might be expected because this would be the time of the recession of the ice sheets, and these would be individuals from populations that are expanding from southern refugia and mixing with other populations and individuals from other refugia. For the black vein white, we find a stark bottleneck in Great Britain around the time of the last glacial maximum, presumably the time it colonized Great Britain. However, we don't find such a stark bottleneck for the common blue. In terms of absolute effective population size, the common blue around the time of its colonization in Great Britain has an effective population size that's a magnitude higher than that of the black vein white. And this could potentially be explained due to differences in genome size, where common blue has twice the genome size of the black vein white. PSMC has limited resolution in more recent times, so to explore the idea of potential population contractions in the 19th century, we looked at runs of homozygosity. Here we have black vein white on the left, common blue on the right. We're looking at proportions of runs of homozygosity in different sizes of runs. So the white bar represent runs of 100 kilobase or greater, and the darker colors represent runs of either 500 kilobase or greater or 1 megabase or greater. Both the British populations or the British individuals for both species show an increase in runs of 100 kilobase greater, which would suggest ancestral inbreeding and would be consistent with the evidence for bottlenecks in our previous slide based on the PSMC reconstruction. However, for the black vein white, we also see a significant increase in runs of homozygosity of 1 megabase and greater, which suggests that there were potentially further potential population contractions in the 19th century. We looked at genetic fitness by trying to estimate genetic load. We did this by taking all variants and classifying them either as loss of function, missense, or synonymous. These are all predicted effects using SNP-F. Here we have black vein white on the top, and we have the common blue at the bottom. And what we generally find, at least for the total number of variants in each of these classes, is that European individuals have higher number of variants for both species. This might not be unexpected because larger populations are expected to harbor more variants, at least in a heterozygous state. Interestingly, although that data is not shown here, it is in the publication. For homozygous missense and synonymous variants, we do find an uptick in Great British black vein white populations compared to the European ones. If we remove the effect of de novo variants and focus only on shared derived alleles, we do analysis of relative allele frequencies. Here, a number greater than 1 can be interpreted as an increase in allele frequency in Great British populations relative to European ones. We have black vein white on top, the common blue at the bottom. And what we see here, at least for shared derived alleles, is that there is an increase in frequencies of missense and synonymous variants in the black veined white. So hopefully, what I've been able to convince you with this data is that the black vein white underwent a severe bottleneck at the time of post-glacial colonization in Great Britain and never really recovered from this initial bottleneck. Additionally, the increases in homozygosity, particularly the runs of 1 megabase or greater, are consistent with a potentially spasmodic rather than a gradual decline in the UK. Additionally, increase in homozygous missense mutations and shared derived alleles may also have made this population more prone to extinction. In the bigger picture, I think our work adds to a small number of recent studies that suggest that metrics of genomic erosion, things like inbreeding, genetic load, could be helpful to monitor or identify threatened insect population or species, even with a small number of individuals. Genomic erosion has been widely applied for vertebrate species, but hasn't been used as much in insects or invertebrates. Finally, there is a lot more work coming out on DNA work with museum specimens, particularly doing population genomics or conservation genomics. But people are using different methods to generate and analyze data. Although our methods are not markedly different, they do add to a body of methods that are widely applicable and easy to implement for insect museum specimens. Finally, we are doing some work on metagenomics to infer whether there's any pathogen infection. However, there are many issues related to working with metagenomics of museum samples. There is quite a high potential here, but a lot of work still needs to be done. If anyone is interested in this, I'd be happy to talk about this more later. Finally, I want to thank everyone that made this work possible. First and foremost, Rebecca, who did all this work, and also our collaborators at Brookes, Professor Jim Shreve, Dr. Casper Breuker, and Dr. Cornet Helns, Jeff Martin at the Natural History Museum in London, and Dr. James Hogan at Oxford University Museum of Natural History. Finally, this work was possible because the Darwin Tree of Life sequenced the genome of the black vein white in 2022, and Konrad Losi, who kindly provided the specimen for that genome.
Hello, my name is Claudia Martin, and I'm a postdoctoral researcher at the University of Edinburgh, working as part of the Darwin Tree of Life project. My work uses population genetic and comparative genomic analyses to study genetic diversity across flowering plant genomes. So today I'm going to outline how I'm aiming to address this question utilising a combination of resequencing datasets and the DToL genomes across native flowering plants. But before I start, I'd like to thank the Wellcome Sanger Institute for funding and my host, as well as collaborators across the Darwin Tree of Life Consortium. So genetic diversity provides the basis upon which evolution can act, and many different factors are known to shape this. But why does this really matter? Genetic diversity itself, not just species as a whole, need to be conserved. Then for species to be successful in a competitive environment, and they need to be able to adapt to environmental change, and genetic diversity allows them to respond to this. This can also facilitate different life history traits and enhance individual fertility and fecundity, which is of course essential for species survival. Having a high genetic diversity allows evolutionary potential and potentially evolution into new species through speciation. And having high genetic diversity, of course, may allow species to avoid inbreeding depression, which is where obviously inbreeding results in negative fitness consequences for individuals. Although we do know some species are able to purge deleterious alleles to avoid these consequences. So this varies widely across different species. For example, we know things such as HIV has a high mutation rate, and this allows host pathogen evolution, allowing this to be very successful. And then at the complete other end of the scale, we've got things such as the pink pigeon, which are critically genetically depleted due to having severe population bottlenecks, resulting in high genetic drift and realized genetic load, which is causing species such as this to be on the verge of extinction. So what shapes this diversity? Well, in short, a combination of evolutionary processes, typically thought of as mutation, selection, gene flow, recombination, and drift. But this is still somewhat a mystery of how these different forces interact. Really, in reality, it's very complex and dynamic and acts over long and short timescales, including, for example, environmental change, different types of selection, such as adaptive and balancing selection, different demographic histories that different species have, for example, through population size fluctuations. And so far, not much research has been done in plants to understand these processes. So that's what I aim to investigate here. So how can we investigate this in abundant species using the Darwin Tree of Life project? Studies do exist in invertebrates, for example, and such questions are asked extensively for specific traits of interest, such as in medical contexts or trait selection in crops or animal breeding. But I aim to ask what drivers of genetic diversity are in flowering plants using widespread and abundant species, and this allows partitioning of the effects of small population size, demographic history from other factors, such as life history and mating systems. Then I also want to look at whether this diversity is clustered by ancestry or not, and how this looks across the phylogenetic tree. So first, I worked briefly with Leanne Haggerty and her team at Ensemble, generating a Busco-based phylogeny. So this is for the flowering plants. At the time, we had many fewer individuals, but this is keeping being updated as more genomes are being released. And the number of flowering plant genomes that are available has doubled since this time last year. So that's fantastic. And a great thanks to Leanne and her team for generating this for me. So this places species with their relative ancestry. And this is kind of a one genome per species approach. As we all know, the Darwin Tree of Life project is generating one genome per species. And this is going to provide that map with which to make comparisons of genetic diversity between more closely and more distantly related species. But many of you may be thinking, but how can we be sure if one genome is representative of a species as a whole? So, i.e., is intraspecific variation consistent across populations? And while it's important to remember that these species are widespread and abundant, the answer to this question just isn't known. So this leads me first to set out to address this question, to provide a baseline assumption with which to address this broader interspecific genetic diversity question. So the core question here is genetic diversity consistent within abundant species? Intraspecific genetic diversity is investigated using a wonderful data set provided kindly by Meng Lu, who's just submitted her thesis at the University of Edinburgh. And this data set includes resequencing of 12 species, or 6 species pairs, each of which are known to hybridize across the United Kingdom. Each population has, each species is sourced from 5 or more populations. So this means that we have individuals across a distribution range within the United Kingdom. Her transects use pairs that are largely sterile through to pairs that are largely fertile. And this is across a range of outcrossing and selfing species, as you can see here. For the purposes of demonstration, here I'll present the results from GM rivali and GM urbanum, one predominantly outcrossing species and one predominantly selfing, which result in largely fertile offspring in the hybrids. So when we look at GM urbanum and rivali, if I look at a genome-wide diversity to produce a principal component analysis for these species that have overlapping ranges, we see that we've got partitioning of the species across the principal component 1 from rivali to urbanum, and we find that our hybrids, in this case F2s, are genetically and also morphologically intermediate. So you can see down here a picture of an F2 hybrid. And so this allows us to answer some questions about ancestry versus life history drivers of genetic diversity. So if we then look at patterns of genetic diversity clustered by species. So if here we see genome-wide heterozygosity as a metric of genetic diversity on the y-axis, and on the x-axis we have our 2 species, rivali and urbanum. And we can see that there's mostly no overlap between these species. They're distinct in their genetic diversity at a genome-wide scale. We see that rivali has a much higher genetic diversity than the pure urbanum, so the pure urbanum are down here and the pure rivali are up here. And actually we have a separate kind of distinguished group of backcrossers. And these are the fertile intermediate hybrid forms here, just on the top right of this plot. So these are the most diverse group, and this is probably due to admixture between the 2 species and hybrid vigor as a result. So the summary of this is predominantly selfing species have a lower diversity irrespective of locality. So it's good to see that we don't have much overlap here between the 2 species. I then wanted to investigate how diversity is distributed across the genome. So runs of homozygosity are regions of the genome that are identical by descent. They're inherited as large chunks and broken down by recombination over time. So large chunks represent recent inbreeding events typically, which might represent small population size, or as a result of closely related individuals breeding, which may happen for other reasons, of course, such as social behavior in vertebrates. And then small chunks of runs of homozygosity typically represent more distant, like breeding between individuals, as these are broken down by recombination over time. So these plots show by Celebos as a Nature Review article, nicely outlining what patterns you might expect to see in different evolutionary circumstances for runs of homozygosity. So here we've got, at the bottom, we've got the sum of runs of homozygosity plotted against the number of runs of homozygosity. And they investigated and discussed the different distribution patterns that we might see. And this has now been used in a lot of research, typically in other wild vertebrate systems. So admixed species, on this second plot here, admixed species have no runs of homozygosity and would therefore be in this far left, bottom left corner of the correlation plot. And at the other end of the scale, we've got bottlenecked populations with contemporary small population size, and they have lots of large runs of homozygosity, typically. So when we look at the distribution of runs of homozygosity between individuals in GM rivali and GM urbanum, we found no long runs of homozygosity greater than 1 megabase in either species, despite a high genome-wide inbreeding coefficient of the selfing species, Geom urbanum. However, we do find lots of short runs of homozygosity in urbanum, the selfing species. But what does this say about life history? So here I've made a similar plot to the previous slide, showing the number of runs of homozygosity segments on the y-axis and the total length of runs of homozygosity on the x-axis. And you'll see that Geom revali, the outcrossing or predominantly outcrossing species, has an admixed history showing in the bottom left-hand corner, all clustering together. And that's also where we can see these F2 hybrids individuals. However, Geom urbanum, the predominantly selfing species, has a distribution, quite wide distribution across this plot, however, never with the high admixed individuals. So I guess importantly, also we see here, despite a variety of results, we see that there's no real overlap between urbanum and revali in these patterns. We then look at the distribution across the genome, and here I'm plotting nucleotide diversity, so another measure of genetic diversity across chromosomes. So chromosomes being on the x-axis here, and yeah, nucleotide diversity measured as pi on the y-axis. And we can also see plotted the histogram of windows. So this shows your distribution of nucleotide diversity windows. So in the outcrossing species, Geom revali, we have a relatively high genetic diversity, and this is consistent across chromosomes. When you then compare this to Geom urbanum, the predominant selfing species, we have a clustering of much lower nucleotide diversity, and across our chromosomes, we can see that there's generally a much lower genetic diversity, including some peaks across the chromosomes of diversity. And so plotted, if you can see the blue vertical bars are each the runs of homozygosity greater than 200 kilobases. And I guess the general pattern that you can see here is, while there are some chromosomes with very big chunks with no runs of homozygosity, actually these small runs of homozygosity are distributed widely across the genome. So in summary, genetic diversity patterns appear largely discrete between species, but this needs to be investigated in more species. And today I haven't had time to show you the other species, but I've conducted this for all of the pairs of species from the resequencing dataset. Selfers have lower genetic diversity than outcrossers, as seen by the homozygous by descent chunks in the genome, and this may be due to a low gene pool. We see that in some cases hybrids have elevated diversity, and this may be due to hybrid vigor. And then going on to future work. So I have some early evidence of the same overlapping runs of homozygosity between individuals across populations. And I want to investigate this further. This suggests that there might be shared evolutionary events, not just shared correlation of numbers and patterns of runs of homozygosity. And then once I apply these questions thoroughly, I want to investigate them at a species level to determine how genetic diversity is shaped. So once we have more and more of the Darwin tree of life whole genomes, I have more power to address these questions. With that, I'd like to say a quick thank you to my wonderful team at the University of Edinburgh and collaborations at the Botanic Gardens of Edinburgh, and a special thank you to Alex Twyford.
Hi everyone, my name is Camille Cornet. I'm a PhD student at the University of Neuchâtel in Switzerland, and today I'm going to tell you about chromosomal rearrangements in butterflies and their causes and consequences. The reason we were interested in butterflies is that, well, Lepidoptera, butterflies and moths are super species rich, like 10% of all described species. But not only are they super diverse, but also they have fascinating genomes. They are holocentrics, which means that instead of having one localized centromere per chromosome, their centromeric activity is scattered all along the length of each chromosome. And this has, in theory, important consequences because it should facilitate the establishment of fusions and fissions of chromosomes. You can imagine that if there's a fission there, on an allocentric chromosome, both parts of the chromosome could be retained, while if it would happen on a monocentric chromosome, like on the left, the upper part that doesn't have the centromere would be lost during cell division. And this, taken together with the fact that chromosomal rearrangement can promote speciation, for example, through decreased hybrid fitness between parents of different chromosomal arrangements, or reduced recombination in the rearranged parts of the genome, well, this together leads to the hypothesis that maybe allocentricity has played a role in the diversification of Lepidoptera. But of course, as usual in biology, things are not that simple, because indeed, most Lepidoptera have actually retained the ancestral chromosome number, despite allocentricity. And only some clades show this huge diversity in chromosome numbers, the karyotypic diversity that is expected from allocentricity. One such clade is Arabia, the genus that we are working on, and they have haploid chromosome numbers ranging from 7 to 52. But interestingly, even within Arabia, this diversity is not equally distributed among different subclades. And in particular, the Tyndaris clade that's highlighted with the arrow encompasses most of this variation. And it's an interesting one because it's simultaneously the youngest, most species rich, and most karyotypically diverse clade within Arabia. And so this leads to the main big questions that I'm trying to answer during my PhD. Why is there this huge karyotypic diversity in some clades but not in others? And what's the link between this diversity and species diversification? So to answer this, it's important not only to understand how fusions and fissions can lead to speciation, but also what are their proximate causes, like mechanistic causes, to understand what went different in the diverse clades compared to the conserved clades. And many things can play a role there. For example, repetitive elements, gene gains and losses, for example, genes involved in DNA repair and things like that. But what I really want to focus on today is the role of 3D structure of the genome and chromatin folding of the chromosomes in the nucleus. So to answer these questions, we've built reference genomes for several Arabia species using PacBio hi-fi reads, but also Hi-C data. So Hi-C data is very commonly used for genome scaffolding, but you can also use this data to get in-depth knowledge of the 3D structure of the genome and compare this among different species. So as a side note, I also just recently got iso-seq data, so long-read RNA sequencing, thanks to a Biodiversity Genomics case study grant that we got recently. And here I want to also wholeheartedly thank Project Psyche, in which we're involved, which is aiming at generating reference genome for all Lepidoptera in Europe in the coming years. And thanks to this project, we have now tripled the number of Arabia genomes that we will have ultimately, compared to in the beginning of my thesis what I thought I would have. And now you can see on the phylogeny on the right all the arrows that are showing the different species for which we have or will soon have reference genomes, and so Hi-C data. So we have a huge phylogenetic coverage of the genus, and here is the Tyndaris clade that I mentioned before. Most species rich, karyotypically diverse, and the youngest within Arabia. So the first thing I did with this reference genome is to build a synteny between the species. So I think this is a beautiful mess. And so you can see the correspondence between the chromosome within each species, going from the most basal species, more karyotypically conserved on the bottom. And on the top, it's the Tyndaris clade that has tremendous diversity in chromosome numbers. You can see here the species with the highest and here lowest chromosome numbers. And I have to say that this is not all the species I have, but it's basically as much as I could fit in one slide. And so I was very happy with that plot, but then I was wondering, what does it actually looks like in 3D? Because of course the genome is not just a linear sequence of nucleotides, right? And it's like complex, folded in very complex ways in the nucleus. So this brought me to ask several questions relating to how this 3D genome structure can be linked to chromosomal rearrangement and to speciation. If you want to know more about the conceptual links between 3D genome structure and speciation, I invite you to have a look at our recent Translational Genetics forum piece where we highlight some of these ideas. But here today, I'm going to focus on several questions that I was asking with this data, such as, do fissions happen more often at the boundaries between 3D features? So by 3D features, I mean like Saad-like structures, chromosome compartments, like these different hierarchical ways in which the genome is folded in the chromatin. And are chromosomes with more contacts in the 3D space, so chromosomes that are closer together in the 3D space, more likely to fuse than chromosomes that are further apart? And lastly, are 3D features evolutionarily conserved along the phylogenies, even through chromosomal rearrangements? So to answer this question, I first built Hi-C maps and for each of the species. And so Hi-C maps, you see the interaction between the different genomic regions as a proxy of the distance. So the most red it is, the higher the interaction and the closer in the 3D space are those two genomic regions interacting. And the blue squares are Saad-like structures. So like places in the genome that are quite compacted on themselves and that it's potentially linked to gene expression. And so I asked if fissions are more likely to happen at the boundaries between these features than elsewhere in the genome. And the answer is yes. Fissions are actually 50% more likely to happen there than expected by chance. And this could be either because of mechanistic explanation that maybe the 3D structure is more loose and more prone to fission there, or my favorite explanation would be that maybe the fissions at these boundaries are less selected against and so more likely to be retained along evolution. For the second question, are chromosomes with more contact in the 3D space more likely to fuse? For this, I also built Hi-C map, but here, in contrary to the previous slides, you see only average interchromosomal contacts for two species. And I asked if pairs of chromosomes that fuse in the sister species have more contact than pairs of chromosomes that don't fuse in the sister species. And the answer is no, there's no difference, which I think is very interesting because it makes me ask, like, then why are those two chromosomes fusing if it's not just simply because they're in close contact? But while answering this question and looking at these Hi-C maps, I also noticed some interesting pattern. First one is not very surprising. You can see very clearly on the left plot that smaller chromosomes have more interchromosomal contacts, like the chromosomes here on the upper right, than large chromosomes on the bottom left. And this is unsurprising because that's a ubiquitous pattern across the tree of life. But interestingly, there are some outliers. The chromosomes that you see here, for example, and the chromosome that you see there, they seem to have high interchromosomal contacts compared to their size. And interestingly, these chromosomes are ancestrally the smallest. And they have retained this high interchromosomal contact property despite going through fusions and other rearrangements. So it seems like there's this evolutionary conservation of the 3D structure. And another way that I started to think about this 3D conservation, 3D structure conservation, is by building 3D models of my Hi-C data. You can see on the left the species with the most fusion, so the lowest chromosome number, and the opposite on the right. And what I think is very interesting is this kind of bean shaped of the chromosome on the left. So it implies that when chromosome fuses, they retain this kind of pearl necklace structure where different parts of the chromosome that fuse keep their like ball shape, but they don't immediately at least evolve like strong intrachromosomal interaction between the different subparts of the chromosomes. Okay, so to summarize, indeed, fissions seem to happen more often at the boundaries between 3D features, but chromosomes that are close to each other in the 3D space are not more likely to fuse than chromosomes that are further apart. And it seems indeed that there is some level of conservation of 3D features along evolution, even through chromosomal rearrangements. Of course, here I have to do a disclaimer that this is very preliminary data, and I am welcoming any kind of question or advice that you may have. And I'm very eager to look deeper into this in the future to see like how conserved are those features, like adding more species from across Lepidoptera to see how deep is this conservation going. And hopefully, at some point, this will help me understand why is there a huge karyotypic diversity in some clade and not others, and what's the link between this diversity and species diversification. With this, I would love to thank everybody that has helped me since the beginning of my thesis. None of this would have been possible without any of them. So especially my group at the University of Neuchâtel, but also Charlotte Wright and Mark Blackster at the Sanger Institute that I had the privilege to work alongside with for 6 months earlier this year. And of course, thank you all for your attention today.
Okay, well, thank you very much, and thank you for our three speakers. What we'd like to do now is have a Q&A session. So could I remind everyone, please put questions in the box that's up on your screen at the bottom. There's a few questions coming in there, but please add them, and we're going to start asking our speakers some of those questions. So if I start off with a question for the first speaker, Saad, are you, with the multitude of Lepidopteran assemblies available now, are you planning on doing similar analyses on several other species to investigate their decline in the UK or Europe?
Yeah, thanks, Peter. We are planning to. So Rebecca, the PhD student whose work I was presenting, has done something similar for the wood white in the UK. That's something we're working on. I would really like to go and explore some of the more the extinct or the extirpated species in the UK. So the Mazarin blue, the large copper. I think I'd love to do that. Again. I'd like to work with some of the museum data just to see how much we can get out of it, how far we can sort of push that. And in this case, at least those species are extirpated, so it'd be interesting to see what we find in their genomes.
Could I follow that up with a related question, really, which is a sort of technical one, really related to how far we can push genomics with museum specimens? Mm-hmm. I mean, it's impressive that you're able to do this from 1 or 2 legs of a museum specimen. Do you think there's a limit to how far, whether all museum specimens of insects will be amenable to this sort of analysis, or are there things that can't be done?
Well, I think for insects, I think this is out in the literature. So the insect specimens are preserved in a variety of different ways. In many cases, the curators don't even know the exact preservation history. And there's a big hit and miss at the DNA extraction step already. And this, I think someone even mentioned this in one of the talks yesterday. This isn't even correlated with age sometimes. Sometimes you'll get perfectly good or perfectly usable DNA from a specimen that's 100 years old, but maybe not from one that's 20 years old. And part of it has to do with, just the lack of information on how those specimens were preserved. When we tried our extraction strategy with the black vein white specimens, some of the most earliest ones, these will be dating to sort of the early 19th century, so from 1820s. When we extracted DNA, we didn't get any we didn't get a readout from the instruments we were using. In this case, it was a qubit HSK. And we didn't do anything with that DNA, but I think you could potentially try and create a library and see what happens with it anyway. At that point, we were just playing it safe and didn't try that. But in hindsight, I feel like you could potentially get something out of it, even if you're not getting a readout with the instrumentation that you have. Okay.
Can I move on to a question? I think for looking at the plants. So one of the measures you looked at there was runs of homozygosity, and you were really using that as a marker of what had happened in the past. Can you speculate on whether that has phenotypic consequences or fitness consequences?
Yeah, good question. So, of course, you mentioned about different lengths of runs of homozygosity, and we know that the longer tracks are more deep in history and more recent, of course, and then the shorter tracks are broken up by recombination events. So I guess, yeah, that leads on to an interesting question about fitness. And I guess there isn't much evidence that very short runs of homozygosity have a clear link with reduced fitness in wild populations. We were somewhat surprised that we didn't find substantially longer runs of homozygosity based on the fact that the F statistics suggest strong inbreeding, especially in these individuals that are predominantly selfing species. So that kind of contrasts to some animal study systems.
Yeah, okay, fantastic. We have lots of questions about Camille's chromosomes as well coming in. So can I turn to some of these? I have what I think this question's really about how much of the Hi-C, how much the Hi-C data can be related to those chromosome maps. Do you need really deep Hi-C data for this, Camille, or something special, or was the Hi-C do it?
With the typical number of reads that we get for genome scaffolding, it's largely enough to have a resolution that allows us to define like TAD-like structures, but to go like deeper at the level of chromosome loops at like a KB resolution, it's not enough, I would say. But you can already have, a good overview of the average conformation of the tissue that you've sequenced for the genome scaffolding. So the idea here was really to be able to reuse this data that is already there and produced by all these sequencing efforts to compare species in their 3D structure. But yeah, of course, if you want to look at things related to gene expression in different tissues, in different situations or developmental stage and things like that, it's not going to be enough to have one representative Hi-C data.
Well, and one thing that I noticed was a little bit surprising to me, actually, was the interchromosomal consistency of because we don't really usually think about interchromosomal contacts, whether they, you sort of have I don't know, there's a naive expectation of sort of randomness there, but that's not what you saw at all. So can you speculate on what's happening there?
The way I like to think about it is like how much a chromosome is kind of loose in the nucleus and interacting with other chromosomes. But it seems, at least in Lepidoptera, that there is really this like chromosomal territories where, as we saw in the 3D model, that like each chromosome is kind of occupying a certain place, at least in how to say, interface, right? Somatic cells. But in other species, there might be more like rubble-like features with like centromere clustering, so like things like that. Of course, here they don't have centromeres, so yeah, like I don't know, but I think it's interesting to see like how some chromosome might be more involved in contact with other chromosomes, but I don't know why. And I think there was a question about like what's so special with that one small, is like ancestrally smallest chromosome, and well, I don't know, for example, what genes are on there for the moment, because also I just got my genome annotation that finished running like this morning, so I don't know yet what genes are in there. But I think something that was super cool with this like ancestrally smallest chromosome, and that was the work of Charlotte Wright, that she showed that the smallest chromosome were more likely to fuse than the bigger chromosomes across Lepidoptera as a whole. So maybe, I don't know what that has to do with what genes might be in there or the 3D structure of this chromosome, but I think that's super interesting to look into that further.
I mean, is your line of thinking there, when you mention genes, is your line of thinking there that there's sort of compartments in the nucleus where there's active and inactive chromatin, things have to be co-localized? Is that the line of thinking?
Potentially, or like co-localized, or maybe like co-adapted, that might have to be co-expressed, or yeah.
Okay. Right, let me just have another look at the question. I had a question for Saad again, right at the beginning, about missense mutations, because it's, you know, we often use them as indications that something has gone wrong, but in terms of fitness. But it'd be really exciting if those could be tracked down to actual functional effects. Is that possible?
So I think maybe at a superficial level, I guess if we take the set of all missense mutations, with the annotation that's available, we could do something like a gene ontology analysis. Obviously, depending on your take on that, that might be convincing or not. It would be very nice to have sort of some idea of the functional effects of those mutations. But at this point, I guess at this point, I wish I was still working with Drosophila, because maybe if I had those lists, right, I could look at information on the at least putative functional effects of those genes. However, even having said that, I think it's still really tricky trying to predict functional effects in an ecological context. I mean, it's such an intangible problem in a way. You have to be able to study it. So I think we have some tools to make some, you know, crude statements. But I think we're still some ways away from really being able to link that, which would be really cool.
Okay, well, thank you all. Thank you. I'm going to thank you all the speakers and for engaging with questions. I'm going to hand over to Kerstin now, who's going to introduce the next speakers.
Yeah, hello everyone. Thanks for staying with us during a really exciting first hour. And I can promise you the next hour will be equally exciting. We have lined up Leon Hilgers from the LOEWE Center in Germany telling us about vertebrate evolution, Amy Denton from the Sanger Institute in the UK telling us about how all these genomes actually get produced from the very start, where extraction happens in the lab and sequencing gets going. And Joanna Meyer, also from the Wellcome Institute, telling us about a massive new sequencing project that has started here that is aiming at sequencing all the moths and butterflies of Europe. So without any further ado, the first talk, please.
Hi everyone, I'm very excited to talk to you today about how we use new reference quality genomes to illuminate turtle evolution. And we've looked at quite a range of different topics, and we'll today only focus on one of them, and that is sex. Sex is arguably one of the most important parts of the phenotype because it dramatically influences physiology, anatomy, and life history. Now, there's different ways that sex can be determined. So on one hand, we have environmental sex determination, where, for example, a temperature cue determines whether a male or a female develops. And on the other hand, we have genetic sex determination. Now, in the course of evolution, species may transition between the two sex determination modes. And if we want to study this, turtles are a great model system. Now, the reason for this is that turtles repeatedly evolved genetic sex determination from an ancestral temperature-dependent sex determination. So here I'm showing you a simplified version of the turtle phylogeny, and there's two main clades, the so-called Cryptodira, which are the hidden neck turtles, and they pull their head back like this underneath the shell. And then there's the side neck turtles, which fold their head to the side, called Pleurodira. Now, most of the turtles that you will think of if you're not interested in turtles specifically are the so-called Cryptodira. I'm also showing you in blue the clades that have genetic sex determination. And you can see that this evolved at least 5 times within the turtles. And there's also 2 main clades, one with a ZW system, so female heterogametic, and one XY system, so male heterogametic like us mammals. Whenever species, turtles or others, transition from an environmental to genetic sex determination, we expect that this affects genome architecture, at least locally. So the best example for this would be the evolution of sex chromosomes, where an autosome gains a master sex determination locus, then recombination ceases around it, repeats, and deleterious mutations start to accumulate, and this may ultimately lead to the death of the sex-linked chromosome, in this case, the Y. So I want to point out that this is a very local effect. It really just affects the sex chromosome itself, but there may be mechanisms that actually go far beyond. And in this case, one example are the turtles. So here I'm showing you again the same phylogeny that I've shown you before, and I've also added chromosome number. Now, the first thing that you can see is that chromosome numbers differ widely across turtles, ranging from around 28 to 68. The second thing you can see is that there seems to be some sort of correlation for genetic sex-determining clades to have rather more than less chromosomes, more chromosomes than the temperature-dependent clades. This has led people to come up with this conclusion, which is published as the title of this paper, named Chromosome Number and Sex Determination Co-evolve in Turtles. And that would be really cool. And this is why the main question of this talk is that actually true? So to look into this, we wanted to approach this from a genomic perspective. And one main hurdle for this is that this entire clade of side neck turtles was lacking high quality reference genomes. So we set out to solve this problem by providing 7 new reference quality genomes. And here I'm plotting for you the genome contiguity measured as the NX in %. So for example, at the dotted line, the N50, this is where contigs of this size or larger contain at least 50% of the entire genome assembly. And our turtles are shown in blue, and the best existing turtle genomes, and I'm not even plotting the worst ones, are shown in gray. And you can see that ours are as good or even better than all of the existing turtle genomes out there. Our genomes are also highly complete, with completeness scores based on BUSCO ranging between 99.5 to 99.9%. So this is based on genome annotations, combining especially multiple runs of our tool TOGA. So now that we have these reference genomes available, we should be able to approach this question that we initially asked, whether sex determination and chromosome numbers co-evolve. But there is actually one remaining problem, and that is the origin of this XY system is actually unclear. Now, what do I mean by this? Let's zoom into this clade, and what you can see here in the so-called Chelidae is that there is a different sex determination systems, at least when we look at the chromosomes. So we have clade with micro sex chromosomes and an XY system. We have a sister clade that has macro sex chromosomes, so very big chromosomes with the Y being even bigger than the X. and of course also genetic sex determination. And then we have a third clade, which is the outgroup to these two, and this has genetic sex determination, but we don't know anything about the sex chromosomes. So the questions this raises is, how many times did genetic sex determination evolve in Chelidae, and were ancestral sex chromosomes large or were they small? And so to approach this, we actually sequenced representatives for each of these clades. So we sequenced Chelidae parkeri, Elsea novagine, and Hydromedusa tectifera. And so with this, we then took an approach of resequencing males and females. And just to remind you, in an XY system is what we expect. Male and female share the autosomes as well as the X chromosome, but the Y chromosome has male-specific sites. So if we now map reads against the female reference genome, as long as the male reads still map to the Y or to the X chromosome in this case, what we expect is to see an increased heterozygosity in the sex-linked region, which is diverged between the X and the Y. And this is increased in the male as compared to the female, and also on the sex-linked region as compared to the autosomes. So this is what I'm showing you here, starting with Elsea, and I'll show you this data twice because we have a haplotype resolved assembly with a very good phasing. And the data I'm plotting is male minus female heterozygosity. So for a sex-linked region, what we expect is an outlier to the top. Now we get one very clear outlier, which sits here on the end of chromosome number 4. And this is great. And by looking into repeats and some data that has been gathered by looking and staining chromosomes under the microscope, I can actually predict which is the X and which is the Y. And also we can have a look at differences in gene intactness, for example, and you can see that genes that are only intact on the X or only intact on the Y also accumulate on the end of this chromosome number 4. If we now add Chelidae parkeri, really nothing is happening on chromosome number 4. Instead, we see something, the highest outlier on chromosome number 25, so a very small chromosome. But interestingly enough, this is actually homologous to the end of chromosome number 4 in Elsea. Now, if we look at Hydromedusa tectifera, again, we find an outlier on a very small chromosome, chromosome number 29, and this is again homologous to the end of chromosome number 4. Now, this indicates that genetic sex determination likely only evolved once in Chelidae, on the stem of Chelidae, namely here, and it should have been a microsex chromosome system. However, we can actually do better and really reconstruct the evolution of these chromosomes by using an ancestral gene order or reconstruction tool called Agora to reconstruct ancestral genomes and trace the evolution of all of these chromosomes along the phylogeny from the last common ancestor of all turtles up to the lineage with the macrosex chromosome, so Elsea. Now, these are shown here. At the bottom, you have the last common ancestor of all turtles, and at the top, we have Elsea. And so we can see that the chromosome, which becomes the sex chromosome in pink, is actually a microchromosome from the beginning and all the way through. And then between Pluridira and Chelidae, so leading up to the yellow lineage, it gains a master sex locus. And then from then on, it stays a microchromosome and only fuses to a macroautosome, namely number 8, on the way to Elsea. So this micro sex chromosome then fuses to a macroautosome, leading to the evolution of a neo-sex chromosome, which is a large one. So in conclusion, this means that genetic sex determination only evolved once in chelids more than 80 million years ago. And despite their age, sex chromosomes remained largely highly homomorphic, especially for those which still have microsex chromosomes. Now, this finding is in line with the more common recent findings that not necessarily all sex chromosomes are born to die. And we also find, of course, that macrosex chromosomes in this case evolved by the fusion of the ancestral microsex chromosome to a large autosome. Now, this brings us back to the overall questions whether chromosome number and sex determination co-evolve. And so here I'm showing you a phylogeny of high-quality turtle genomes within our data set. And I'm again showing you the origin of genetic sex determination as this little double helix. So we have two cases here. One of them we narrowed down. This is the one on top for the Chelidae. So what we now do again is reconstruct chromosome evolution, and we add up all the fissions and fusions, and we get ancestral chromosome numbers and reconstructions of the entire chromosome. So for example, here for the last common ancestor of all turtles. Now I can plot the number of chromosomes on this phylogeny, and what you can see is that we mainly have one major change, and that is a reduction of chromosome number on this clade here, including Pilomedusa and Peltocephalus, Podocnemis, and Erimnochelis. Now, actually, after the evolution of genetic sex determination, surprisingly little happens. And this doesn't really change when we look at the speed, the rate at which chromosome level mutations occur. So here we see it's pulsated, so there's few branches where a lot of things are happening, whereas most of the time nothing happens. And then this doesn't really correlate with the evolution of genetic sex determination. And this also doesn't change if we look at fusions and fissions separately. Now, I said before that chromosome evolution is pulsated, and this is in line with recent results on butterflies from the Baxter lab. And I can also show you here what this looks like in turtles. So here we have a lineage that evolves very slowly from the last common ancestor of all turtles to Dermochelis, so around 250 million years of evolution. And all that happened in those 250 million years are 4 chromosome fusions. So this is very, very little chromosome level mutations, but all of them occurred on a single branch. On the other hand, we have lineages where much more happened, like going from the last common ancestor, so same age of all turtles, to Peltocephalus. And here you can see that we really see more than a 10-fold increase as compared to the leatherback. And again, the changes seem to accumulate on only a few branches, which indicates that there's processes that kind of destabilize the karyotype. There's a lot of follow-up evolution happening. And so this sped up, and then it slows down again when it reaches an equilibrium and stays more and more stable for a while. Lastly, I want to point out one thing, and that is that these new methods allow us to see processes that would be hidden to traditional methods. So even if we correctly reconstructed that the last common ancestor of Podocnemidae had 15 chromosomes and Peltocephalus has 13, then that will lead us to inferring 0 fissions and 2 fusions, because that would be most parsimonious. But what actually happened is that we see 9 fissions and 11 fusions. So that would actually lead to a 10-fold underestimation of the amount of chromosome level mutations that actually happened. So this means that these new methods are really useful to really reconstruct chromosome level mutations. And with that, I want to conclude this talk. So what we find is that in contrast to existing hypotheses, chromosome number and sex determination do not co-evolve. And we see that chromosome evolution is pulsated, which may be linked to repeats, but actually I don't have the time to talk about this today, so we can happily discuss about this later on. Now with that, I would like to thank all my colleagues and collaborators, especially the Hilgers lab, as well as my funding bodies, which allow me to do the research that I'm so passionate about. And so with that, I would like to thank you all very much for your attention, and I'm looking forward to your questions. Thank you very much.
Hello, my name is Amy Denton, and I'm an Advanced Research Assistant in the Tree of Life Core Laboratory, and I'm presenting on behalf of the team today to provide an update on the improvements that we've made over the last year. So as you all know, the Tree of Life programme generates reference genomes for a vast array of species for several major projects, and these include the Darwin Tree of Life project, Psyche, the Aquatic Symbiosis Genomics, the Vertebrate Genomes project, the European Reference Genome Atlas, Ancient Environmental Genomics Initiative for Sustainability, and these all come under the Earth Biogenome project. And it's the Tree of Life Core Laboratory who provide the wet lab and sample processing support for all of the work within the Tree of Life programme. So we help support all of these different projects. And there are currently 13 of us in the team, and we all have different areas of expertise, whether that be specific taxonomic areas or specific processes. But we all work together and perform these processes, which cover sample preparation and homogenization, high molecular weight DNA extractions, high molecular weight DNA fragmentation, fragmented DNA cleanup, and RNA extraction. And as you can see here on the right, these are all the different protocols that we have for these processes. And this here is a link to our protocols IO collection. And it was last year that our senior scientific manager, Caroline Howard, presented the wet lab processes within the Tree of Life core laboratory, which have helped enable us to deliver 1,000 genomes for the Tree of Life program. And this talk also included an introduction to our workspace on Protocols IO, where all of these wet lab processes are documented as protocols which are freely accessible. However, a lot's changed in the last year. So whilst last year, Caroline was discussing how we'd help to deliver 1,000 genomes for the Tree of Life, we're now actually very close to 2,000 genomes being released, and we have over 2,600 genomes assembled. So what have we done in the last year that's helped us to contribute towards almost 1,000 additional genomes? Well, first of all, we've developed new protocols which have been published onto Protocols IO. So we have the updated automated plant organic extraction, which has been developed by Ben Jackson. We have the modified Omega Biotech eZNA protocol and the automated MAGATRACT for small arthropods protocol, which has been developed by myself. And we have the automated nucleated blood nanobind protocol, which was developed in collaboration with PacBio and by Adam Bates. And hopefully later this year, we will have a new version of our protocol collection, which will include these new protocols. But if you do want more information on the modified Omega Biotech eZNA protocol, then please do look out for Graham Oatley's ASG presentation on Nadaria in this conference. And as well as new protocols that have been published, we've also got new protocols which have been brought into our routine work in the lab. So we now have PIMS, which stands for Picogram Input Multimodal Sequencing. And this has been developed by Chris Lama with Erna King, Vitek Morik, and Adam Bates. And it's been designed for the sequencing of myofaunal animals from single specimens. And it's been successfully applied to nematodes, tardigrades, rotifers, and flatworms. So the tardigrade data now covers 30 sequenced genomes, and the nematode data now covers 70 sequenced genomes, which means we have now 100 PIMS produced genomes. But in addition to myofauna, we're now looking at using this protocol for very small arthropods, and we've currently processed ostracods and mites, and in the future, we'll be using it to process aphids. And we've also done lots of R&D over the last year with lots of developments into various taxonomic areas. So one of these has been corals. So traditionally, coral extractions have given very poor results. We've had low yields of severely genomic degraded DNA, with the majority of that being low molecular weight whenever they've been processed routinely using the cryoprep to disrupt and the MAGATRACT extractions. But we found that by disrupting coral samples via bead beating using the FastPrep 96 and combining this with organic extractions, that there's been success for several species of corals with a increased quality and quantity of the DNA obtained, as you can see here for this species of stony coral. So this was when it was processed routinely, a very poor yield and poor profile, whereas when bead beaten and given an organic extraction, a much improved profile and yield. And this work has been undertaken by Ben Jackson, who has applied his knowledge of bead beating on organic extractions in plants onto coral species, which really needed help to be passed through our pipeline. And if you'd like to know more about coral samples and the R&D that's gone into them, then please do watch Graham's ASG presentation on Nadaria. And we've also had lots of R&D into plants, especially recalcitrant plant species. So Ben, our taxonomic specialist for plants, found that a pre-lysis sorbitol wash could be introduced during the extraction process to help remove contaminants. And by combining this sorbitol-induced plasmolysis of plant tissue homogenates alongside the automated PO protocol, both the DNA quality and quantity could be improved. So SOPO, as we refer to it, has been particularly helpful in the processing of this sample here, Calluna vulgaris, which is the common heather. And so as you can see here, when processed with the regular PO protocol, we had a very low yield and the sample was very contaminated. Whereas when processed using SOPO, the yield was dramatically increased, the quality was improved, and the contamination also drastically reduced. And this enabled that sample to be sent for sequencing and completed this species genome. And if you'd like to know more about sorbitol washing, then please do look out for Ben's BG24 poster. And we've also had R&D for bryophytes, as these plants we'd not been able to process routinely in the lab due to their low tissue mass. So as a result, a low input plant extraction protocol was developed to enable their processing. So LOPO is a modification of the automated PO protocol, and it's been optimized to maximize the DNA recovery of bryophytes that range from 1 to 15 milligrams in weight. And these modifications involve adaptations to the bead beating process, sample lysis, and the use of a vacuum concentrator. So for this example here, we've got the flagellate feather moss, and when processed with LOPO, we got this extract here that was just under 250 nanograms of DNA from just 6 milligram input of tissue. And this was then processed using the ULI library prep to give us this sample here, which when sequenced on one cell of the Revio gave us a yield of 67 gigs and enabled this sample to be, the genome to be completed and assembled. And now, because of this, lots of other bryophytes will be processed using this protocol. But it hasn't just been plant samples where the tissue availability has been limited and we've needed to deviate from our standardized protocols. So in this case, we've had to alter our power mashing protocol for the Podarcus wall lizards, where we had only the tails provided for genome generation. So the tails were too small for the cryoprep, but if we tried to power mash in the regular way, they would have been too tough. So had to alter slightly, and the samples therefore had an initial incomplete disruption using the power masher, just to break the tissue slightly. They were then allowed to lyse for 1 hour to get that lysis buffer into the tissue, and they then had a manual disruption using the power masher pestle to fully break down the tissue, and given a further 2 hours of lysis to really let all that DNA come out into the lysis buffer. And the samples were then extracted using the manual MAGATRACT protocol to really maximize the amount of DNA we could recover. So for this example here, we have the Catalan wall lizard, and here we had just 4 milligrams of tail inputted into the manual extraction. However, we managed to recover just under 2,000 nanograms of DNA. And this was processed using our routine shear and spry to give us this sample here. And this was submitted to sequencing despite the retention of the 2 kb peak, as actually when it went for further library prep, there was a modified spry that removed this. And this sample achieved 55 gigs on one cell of the Revio and allowed data completion. And if you want more information about the Podarcus wall lizards and examples of other chordates that have needed help for a pipeline, then please do look out for my BG24 poster. But it hasn't just been the development of new protocols and processing difficult samples that have enabled us to increase the number of genomes we've completed. We've also been working to ensure that samples submitted for sequencing reach full coverage. And this has been done by initiating a top-up management process, which has been especially important for small specimens where we're unable to perform repeat extractions in the lab or provide more DNA for sequencing. Initially, the time of storage, it was a concern that that could be a potential issue. However, this ULI library here for the mintleaf beetle shows that a strong yield could still be obtained 9 months after the library was made. So here in July of last year, The library was ran on the SQL and got 19 gigs. And then the remaining library in the top-up process was found and ran in April of this year, and it was still able to achieve 22 gigs. And this top-up management process is also compatible with LI libraries. So in this example, we've got another small species, the orange-spotted Scaphidium, and this reached data completion after a top-up. And again, the library and its longevity could have been a concern, but here this same library gave us a strong yield 10 months after it was made. And this library also crossed a platform boundary. So as you can see, the initial sequencing was in August last year on the SQL, where it got 28 gigs from a full cell. And the top-up was a multiplex sample, which was run on the Revio on May this year, and it got 29 gigs from a quarter of a cell. And finally, we've had improved data sharing and transparency with the ability to track samples through various pipelines and across multiple platforms. And this is thanks to the Tree of Life portal, which has been developed by the ToL team. And for the core lab, this has helped us to track the extractions and sequencing results by finding them all in one place due to ToL portal pulling in data from various different sources. And it's also aided in our top-up management process as we're able to find out if there's any library remaining for samples, and so then we can request them for repeat sequencing. So with that, I would just like to thank everyone involved in the Tree of Life programme. I'd like to thank all of the Darwin collaborators that you can see here. I'd like to thank all of the Tree of Life teams. I'd like to thank Sanger Core Facilities, particularly SciOps, and especially their long read team and R&D team. And I'd like to thank all of the ASG, VGP, ERGA, and all of the other collaborators and collectors for the Tree of Life programme. I'd also really like to thank the Tree of Life Core Laboratory for all of their work and for being able to present it on their behalf. And I'd like to thank you all for listening. Thank you very much.
Hi everyone, I am Joanna Meyer. I'm a Royal Society URF and group leader in the Tree of Life program at the Wellcome Sanger Institute.
And hi, I'm Charlotte Wright. I'm a PhD student at the same institute.
It is our great pleasure today to introduce to you Project Psyche. This is a project that started about a year ago. It is led by Mark Blackster and the two of us. And the aim is to generate and explore reference genomes for all 11,000 species of butterflies and moths of Europe. So we aim to sequence the genomes of all members of the order of Lepidoptera that occur in Europe. The first phase is funded through Wellcome via the Tree of Life program at the Wellcome Sanger Institute. You may wonder, why is it named Project Psyche? And that's because the Greek goddess of the soul, which is called Psyche and depicted here in this relief, it's often depicted with butterfly wings. And you can also see it here in our logo, which was designed by Charlotte. So why, if we want to do such a mega project, would we pick Lepidoptera as our taxon of choice? Firstly, they are very important agricultural pests. They cause damage in large amounts, and not just to our crops, but also to our clothes, as you might think right now. But they're also extremely important for the ecosystem. For instance, they're very important pollinators, also for our crops. They're important food sources, for instance, for birds, reptiles, spiders, and many more. And they're also important biodiversity monitors, so they can be used as indicators to assess how insects and thus the entire ecosystem is doing. They are also quite feasible for such a project of doing genomes, because genomes of Lepidoptera actually assemble very easily. These are 4 examples here that even before curation, you can see their chromosomes as neat little squares, which indicates that with just PacBio and Hi-C data, the reference genomes were basically assembled on their own without much manual work. So this is much more feasible for the order of Lepidoptera than for some other taxa where the genomes are highly repetitive and very difficult to assemble. Now, having lots of reference genomes for many, many species in the same order gives us amazing opportunities to address lots of different questions. Like, for instance, how do chromosomes evolve? And this is something that Charlotte Wright and Mark Blackster in the paper here on the bottom have already looked into with a small subset of 200 species. Or, for instance, how do opsins evolve? How do butterflies and moths see the world? This is something that Peter Mulhare and Peter Holland in Oxford have already looked at in the paper here shown on the right. And then we can also look at many more other things like the genetic basis of migration, what regions are under selection, how do we figure out what are the predictors of genetic diversity, and many, many more questions. And reference genomes can be the base for all of these studies. So this is why we do it. Now I will hand over to Charlotte, who will talk to you about how we do it.
Yeah, so we're aiming to subset this project, which is very ambitious, into 3 discrete phases. We're in that first phase at the moment, which is to generate 2,000 genomes. And during this phase, what we're really trying to hope is to get taxonomic diversity, so sampling all known superfamilies and all families that have members within Europe. And we're doing this by having samples from key sampling hubs and with the sequencing performed at the Wellcome Sanger Institute. In the second phase, we're branching out to 5,000 species, and with this, we're hoping to get whole ecosystem sampling for selected ecosystems. But really importantly for this phase, what we're hoping to do is to widen sampling and also sequencing participation. And so that will involve establishing sequencing hubs outside of the UK across Europe. And finally, in phase 3, we'll try to get as close as we possibly can to all 11,000 species of Europe, and therefore completing all those that we find in Europe. So like I mentioned, we're currently in phase 1. And so for this, the sequencing is being performed at the Sanger, and this work is being led by myself, Joanna, and Mark Blackster. And our sampling hubs are shown here. They're distributed across Europe, including countries like Italy, Spain, and Switzerland. And they are led by an amazing team of researchers that you can see on this slide. And these are the people that really are helping to make Psyche a reality by going out and collecting these amazing species from across Europe. Alongside this, what we are really driving to do and making great progress in doing during phase 1 is to build a community that spans from researchers involved from collection to analysis to applications to conservation agriculture. And to this end, we have over 100 researchers involved in Psyche, including representatives from over 20 countries, which is really exciting. And so together, what we're already doing is developing standardized protocols. So for example, here we're showing the standard protocol we've developed to sample lepidopterans in such a way that we can retain that DNA to do these cutting edge types of DNA sequencing. And I'll now pass back to Joanna, who will tell you about what collections we've been up to this year.
Yeah, so the sampling hubs have been extremely successful. This year, the 7 hubs have already collected over 1,000 species with lots of different fieldwork trips involving lots of different people, which is really amazing. And so when they're in the field or then back at the lab, they first collect the specimens and then take standardized photos. And when the specimens are still alive, and then after killing them, they dissect them. And for larger moths like this one here, the head goes in one tube, the thorax goes in another tube, and the abdomen in a third tube. And this is because we use those different body parts for different sequencing strategies. So we can get the genome and the transcriptome from the same individual, ideally. And the wings are stored in envelopes and kept as vouchers. And then in addition, they also record all the metadata for these specimens. So we have exact GPS coordinates, information on the sex of the specimen, etc., and keep good track of who collected it, who identified it, and so on. And this is then the tubes then come to the Wellcome Sanger Institute in phase 1, in later phases, hopefully to lots of different places. And there we assemble the genome. Like for instance, this one has 31 nuclear chromosomes, but in addition to the nuclear genome, we also get its mitochondrial genome. And in the case of this moth, 3 additional genomes for free, which are from Wolbachia bacteria that lived in this specimen. So we get all the DNA out and assemble all the genomes that were present in that specimen. And once we have the genome, we publish it openly for everybody to use for any question you're interested in. And then we also publish a genome note, which is a small publication presenting the genome, why this species is important, quality metrics, etc. And this is also very important to recognize the collectors and the people who contributed to writing this publication. So now I will hand back to Charlotte, who will tell you about where we are at.
Yeah, so despite Psyche being a relatively young project, we've already made really exciting progress. And so at the point where this talk was recorded in September, a total of 915 genomes were in progress or entirely finished through Psyche. And of those, around 600 are publicly available and finished all the way through. And because of this great progress, we think that it's really realistic to get to 1,000 genomes by the end of the year. And you can see that milestone with the green star there. And with this rate of progress, and we hope to scale up as well with additional samples from the collection hubs that have been so active recently, we'll make getting to 2,000 very realistic for the end of 2025. And I should say that while a lot of this progress is now being driven by Psyche, we've also very much benefited from the Darwin Tree of Life project, which is sequencing all the animals, plants and fungi in the British Isles, and has also generated a lot of lepidopterans, which is helpful to Psyche as well. And so to put this remarkable progress into context, really, you can see this graph here, which shows the total number of species that have been generated for each of the projects involved in the Earth Biogenome Project, which is the umbrella project for lots of biodiversity genomics. And you can see Psyche is right up here, second only to the Darwin Tree of Life project, which is really amazing given that this is just the taxon Lepidoptera and shows what excitement there is really about this project. So with that milestone coming up of the first 1,000 genomes, that presents a really exciting opportunity to the community to start to use these genomes to make scientific discoveries. And so to this end, we're going to have Psyche community working groups that are going to begin to tackle different questions that can be answered with these genomes. And topics that are of particular interest at the moment include things like phylogenomics, understanding how the species are related, looking at cobionts, exploring chromosome evolution and key processes like speciation, and also applications like applying genomics to conservation and pest control. Not only are these going to be deep dives into biology, but also we hope that lepidopterans are going to lead the way for generating standardized resources as part of the genome afterparty, including things like whole genome alignments and protein morphology predictions, as well as knowing where ancestral linkage groups are across the breadth of Lepidoptera. Psyche, as Joanna mentioned, is really laying the foundations for other research, and by making these genomes publicly available that are the starting point for things like population genetics. And so to this end, a really exciting separate initiative has been established by Patricia Valverde and Chris Wheat called LOEWE, European Lepidoptera Populations of Genomics Consortium. And what they are aiming to use the Psyche genomes to then generate population genetic data in order to really understand regional genetic diversity, inbreeding and local adaptation of LEPs to have a better understanding of how to inform biodiversity management in Europe. And again, this is very much a consortium with sampling hubs across Europe and a really exciting project that's been enabled because of Psyche. Because of all of this activity in both the reference genome space and population genetics space, Nicholas Wahlberg successfully applied for a cost action together with a group of researchers that is called LEP10K Genomes, which is supporting the community of researchers to get together and use this data and work up best practices. And so to this end, there will soon be funding to organise workshops and training courses, as well as enabling research exchanges for early career researchers, as well as also working with external stakeholders to identify societal needs that genomic information can help with. And so this is something that very much will cover both Psyche and LOEWE to enable us to do best practices and get the community together to analyse these things in a collaborative way. So the Psyche community is very much growing, and we're looking forward to more people joining us as we develop. And so if you're interested in getting involved, we have monthly Zoom meetings, and we have a mailing list that's used to coordinate this, as well as our Discord server. So please do either go via the website link or sign the QR code if you're interested in getting involved in any aspect of Psyche. And you can also see more via our website. Thank you very much for listening.
Hey, everyone.
Hello again. We are live again now for our question and answer session. And firstly, I want to congratulate you all to 3 really fantastic talks. And there are lots of questions in the Slido. Please type in more if you have any. So the first question is from Eric Jarvis, and he asks whether we might have missed out on sex chromosomes in the turtle and tortoise genomes that are already existing within the VGP. I checked internally, and apparently what we've produced so far was mainly temperature-controlled sex determination. But Leon, do you think we have missed out a few?
No, this is exactly correct what you're saying. So what the VGP has sequenced so far is all temperature-dependent species, as far as I know. And there were, however, 2 cases from the very earliest species where actual chromosomes are labeled as scaffolds. This is something that I found when I traced chromosome evolution across the turtle phylogeny. So this is something that I've also gotten in touch about, and I think it's already been corrected or going to be corrected, I think.
Fantastic. Staying with the sex chromosomes, Peter asks what that really means for recombination if the small sex chromosomes are homomorphic.
So what does that mean for recombination? Generally speaking, microchromosomes have higher recombination rates. The fact that they are or appear very homomorphic can have multiple causes. Of course, that can contribute. Then secondly, there is a possibility, as far as I know, that you can get sex reversed individuals. I don't know how often that occurs. We have by far not enough knowledge about that of these species in nature. But that could help to maintain a certain degree or increase how homomorphic these are. Ultimately, we have not been able to narrow down the master sex determination gene or the specific, like a very tiny window where this should be encoded. So with regards to that, I'm guessing that it's fairly small, but for the non-recombining region, it's not massive, which is also why other people have not been able to do it in the past with somewhat less resources, to be fair. Yeah, so that's what I can say about that. And finally, one thing is, we can only get a limited amount of individuals to do these analyses. So this kind of limits our resolution to some degree as well, because these species are really rare on the, say, reptile market. So people that have them, zoos that have them, they are fairly protective of these individuals, and they just don't want to share a lot of individuals for us to poke a needle inside their neck. So that is may also contribute to the fact that we don't see as much of an outlier window as we might be able to if we narrow it down further if we had more individuals.
You mentioned sex reversal is possible, which reminds me of fishes, where you sort of find everything and anything and all at once. Are there any lessons to be learned from how it works in fishes? Are there parallels?
Oh, I love fishes, so surely. No, but so I guess for turtles, we don't know enough how exactly they do it. What is probably the best investigated thing with regards to sex determination is temperature-dependent sex determination now, but also in the other half of the turtle phylogeny. And we've been looking at that and how that evolves following genetic sex determination. There seems to be something very interesting going on as well. There are almost certainly, based on chromosome homology, different, as far as I can see so far, it's going to be different master sex genes, just like in fishes. But the diversity that we have across all of the fishes, that is not paralleled in turtles. Per species, however, like in living species, these are only like 350 or so, if I'm not totally mistaken, in turtles. There is an enormous diversity if we consider that. So 5 times independent evolution of genetic sex determination is pretty remarkable.
And the next question for you is whether you are looking into temperature control sex determination in conjunction with the GSD in turtles as well.
Yes, I mean, to the degree that I want to know what happens to the system so as far as we know, the last common ancestor of all turtles, I would say the majority of evidence really suggests that this was a temperature determining species. So what I'm looking at right now is the evolution of this system that was probably ancestral. What does that do once genetic sex determination takes over? And I think there's interesting results there as well, but it will go beyond this this round right now, unfortunately.
Thank you very much. Let's get over to Amy's talk. So there are several questions here as well, and people wonder what really is the challenge that is still out there. What is the trickiest bit? What do you really need help with? And also, how do you get that help? How do you interact with other people nationally and internationally in order to tackle those issues?
I guess some of the challenges are, I guess, sometimes the mollusks can be an issue. They don't like to extract very well, or sometimes we do get the DNA out and then they don't want to sequence well. Isopods can sometimes be challenging. They're very small, but then have relatively big genomes, and sometimes they work with our routine extractions and sometimes they don't. So we're doing a lot of R&D into those as well. And then crabs and shrimps can sometimes cause a bit of an issue, but hopefully bead beating might solve that and just help with the disruption to allow us to get that DNA out. I think a lot of it is talking to people in conferences like this. We have discussions with PacBio as well to help with any possible sequencing related issues. And yeah, we've got our workspace on Protocols.io where other people externally can join, and people are free to email us as well. And yeah, we just discuss lots of things between one another.
You've already nearly covered two more questions. One was, how can people get to your protocols? And you said they're all available on protocols.io. And also people want to know, especially Aporia wants to know what the best practices are for extraction of crabs and shrimps, which you just mentioned.
Yeah, we're still working on possibly the best, but hopefully maybe bead beating might help out a bit. Some of them do seem to be okay with our routine magatract extractions, but others we do seem to struggle to get that DNA out of them in the first place. But hopefully we'll be doing more R&D into them in the next year or so.
Excellent. There are also questions for Joanna and Charlotte. Charlotte is here with us today to answer them. So Peter asked whether there will be clean genomes possible from larvae, or do they have to be reared to adults first?
It's a good question, because rearing to adults can be really challenging and time consuming. I think at the moment the general idea is still adults is really, really preferred, because larvae, although they've got the cuticle, most of the rest of it is the leaves or whatever they've been nibbling on. And so you're actually sequencing a lot of plant rather than a lot of the individual. But I think it's an interesting question and something we do need to think about, because it's going to be really hard to get adults of some of these things. And I wonder if with new technologies that are like the PIMS method of like ultra low input methods of DNA, there could be a way of kind of testing if we just took like the heads of larvae and use that as input. Maybe that's the way to go. So try to really specifically get the body parts that are less likely just to be plant is one option, or at least reduce the need of multiple adults, just have one and to use ultra low input methods maybe. But yeah, larvae are hard.
Good luck. I wondered, what do you think will be the first question that a large project like Psyche will be possible to answer?
Really good question. There's so many things. It depends who you ask and what they're interested in. But I think one of the really things that didn't come to mind for me, but first it's a good opportunity to know what are the Lepidoptera of Europe. That's something that I thought would be a simple case of find a list, but there is not one species list of what are the Lepidoptera of Europe. So that's a quite low hanging fruit first thing. And obviously with genomic sequences, you can actually interrogate what we currently think are different species. Are they actually different, and what's
the kind of the cryptic variation that actually exists and diversity we didn't know about.
Thank you very much. So there are no more questions in the Slido. So I'm hoping that we can bring everyone back into the Zoom room here, because maybe you have questions for each other, and we can spend the last remaining minutes in order to address them. But everyone in the Zoom room can already start if you have questions for each other. If not, I'm waiting for Camille, who might be able to answer, despite not currently being here on display. We all love us a good, excellent. We all love us a good Hi-C map. And I wondered whether you have any plans, you briefly mentioned this earlier, but I wondered whether you had any plans on creating Hi-C for on a population scale, basically, in order to identify happenings in the same population where you can see chromosomes fusions or fissions, and maybe even hybrid zones or something like this.
I would really love to do that. Unfortunately, we are not going to have enough money to do more Hi-C. So it's only going to be one Hi-C dataset per species. But I think that already we can do some pretty cool things, like looking, for example, at sister species that have a lot of rearrangements and looking at how are the rearrangements affecting the 3D structure of the genome? Like if the rearrangement is kind of keeping the 3D structure intact, or that are those rearrangements more likely to be conserved and retained, or are some rearrangement like really messing up with the 3D structure, like TAD-like structure? This is really something that I would really love to look into. And we do have some sister species pairs that have a lot of rearrangements that we can look into. Yeah, so, but I would love to have also population Hi-C data, but not there yet.
I'm not aware of anyone actually doing this. Would that be a first? Or am I just not very well informed?
I have a friend who's doing Hi-C data on different strains of pathogenic fungus of wheat. Yeah, so she's comparing the 3D structure on the different strains and if it's pathogenic or not.
Any more questions? Amy, what's your, sorry, Charlotte, go ahead.
By chance, I should know, but any of the individuals that have been sequenced, are they heterozygous for a fusion or a fission?
Actually, I just got an email a few weeks ago from the curation team at the Sanger that told me that one of the species that we sent for Psyche is actually heterozygous. So not in the species that I have sequenced myself, but there are some that are either in curation or very recently released that are heterozygous. Yes, it's very exciting.
Leon, if I can maybe come back to Eric's earlier question. Do you see differences in quality between the turtle genomes that you produce and the turtle genomes that we've produced for the rest of the VGP?
Not, at least not massively for the recent ones. So I'd say there's a slight decrease in the very early ones. And then we, in the course of what we've done, so by the way, we also contribute genomes to the VGP. So the line is more and more blurry, I think. But yeah, the very early ones, the clear reads, and then now that we've moved on to newer technologies, I think the most recent genome that is another step ahead, as you'd expect. This is also where we have this really high quality phasing, and we can then look at the X and the Y, and so this one is a really nice one.
Could I ask? I have a question about the black-veined white butterflies. I'm going back to Saad's talk right at the beginning. So, do you think now that we have genomic information from, extant European populations and from extinct museum specimens, that this sort of information can be helpful for reintroduction strategies. I mean, that would be a real sort of flag for genomics if successful reintroduction of species was based on the genomic information.
I think so. Not from what we've done, but I think with things like the LOEWE, if we get more information about the black grain white across Europe, that might be informative. I guess something, if we want to use genomics to inform reintroduction, something that we might be looking for is obviously trying to maximize genetic diversity. That might be one tool. Looking for maybe environmental and genetic associations. So picking populations maybe that have similar climate or habitat to the UK. I think it could be really useful for that kind of stuff. Also, I think it might be important if we are picking populations or from multiple places, screening them for endosymbionts like Wolbachia or Spiroplasma will also be useful, because we could end up with a situation where we have two populations which might be carrying different Wolbachia strains. We could get a potential case of cytoplasmic incompatibility. So yeah, I think genomics has a lot to offer for reintroduction purposes.
Right.
There was a lot of coverage on butterflies. I would like to ask more about plants. Claudia, what do you think will be most helpful for your studies in terms of reference genome creation, additional data acquisition, etc.?
Yeah, I guess the main thing is the rapid takeoff of number of genomes that are available. So I guess when I started to ask these questions 11 months ago or so, we had 74 genomes, I think, available. Now there's well over 200. So that really allows me to ask these questions at a finer scale than before. Yeah, so that's the main thing, the numbers, to start to draw together these patterns across the phylogeny. But yeah, I guess that's why I was trying to pair it with this free sequencing data so that I can ask them at fine scales and at broad scales.
Ms. Loma asked online for Camille whether she thinks that 4C would make a difference to research.
Actually, I've never heard about Porcy before, so I'm looking forward to look into it. But so yeah, I don't know how to answer this question. Thank you.
Okay. Are there any more questions? If not, then I would like to thank you all for a fantastic session and give everyone else a 2-minute breather before the keynote session that is following on this one. Oh, do we have Davos first? Is it the musical session first? I should know this. No, it's the keynote first. So please, everyone, look forward to this. And thanks very much again to everyone, and applause for all your presentations. Yeah. Enjoy the rest of the conference.
Yeah, thank you, everyone.
Goodbye.